In this video lecture, we will discuss about elevated temperature test, which is also known as Eichmann test, This elevated temperature test, or we can say that Eichmann test, it is mostly used to detect the fecal coliform in the water sample. This test is mostly used to detect the fecal coliform, is it is present in the given water sample or the not. To perform the Eichmann test, we required water sample, which is to be tested, is it in the water sample there is the fecal coliform is present or not. And second most important thing, we required sterile McConkey lactose bile broth, or we can also take BGLB broth, that is brilliant green lactose bile broth, anyone we can take. To perform the Eichmann test, we required water sample and we required McConkey lactose bile broth, or we can also take BGLB broth, that is brilliant green lactose bile broth. Before we go further, we should know about the composition of the McConkey lactose bile broth and BGLB broth. McConkey lactose bile broth, it is consist of lactose, peptone, NaCl, crystal violet, bile salt, neutral red, agar powder is in that it is a broth, and the pH we maintain around the 7.4. Okay, this is the composition of the McConkey lactose bile broth, which is consist of the lactose, which is a sole source of carbon, peptone, it will provide the multi-nutrient, NaCl will maintain the osmotic pressure, crystal violet and the bile salt, this both crystal violet and the bile salt, that will inhibit the gram positive bacteria as well as the fastidious gram negative bacteria, that will inhibit the gram positive bacteria and fastidious gram negative bacteria. Neutral red, it is an pH indicator dye, which is the main component of the McConkey lactose bile broth, and that is we maintain the pH around 7.4. This is what the composition of the McConkey lactose bile broth. We should also know about the composition of BGLB broth, which is brilliant green lactose bile broth, which is consist of peptone digest of animal tissue, or we can say is that peptic digest. Of animal tissue. Then there is the lactose sugar, ox bile, brilliant green, and we maintain the pH. Okay, this is what the composition of the brilliant green lactose bile broth. Peptic digest of animal tissue, that will serve as a source of many essential nutrients. This will serve as a source of many essential nutrients. Lactose, it is a carbon source. In this, the organisms will ferment the lactose sugar into the acid and gas. Then there is the ox bile, that will inhibit the gram positive bacteria. It will inhibited by the ox bile. Then there is the brilliant green. It has a two function. It will act as a pH indicator dye and it will also inhibit the fastidious gram-negative bacteria. So this is what the composition of the BGLB broth. It is consist of peptic digest of animal tissue, lactose, ox bile, brilliant green, and we maintain the pH. To perform the Eichmann test, or we say that elevated temperature test, first of all we will take the McConkey lactose bile broth. This is what a McConkey lactose bile broth original tube, or we can also take brilliant green lactose bile broth. Okay, any one we can take. If we are taking the McConkey lactose bile broth, first of all we will take this, and in that we will add the 1 to 2 loop of the water sample. Okay, and in this tube already there is the Durham's vial, it is present. Durham's vial we have already inserted in this tube. Okay, first of all we will take this tube and we will add our water sample. After adding the water sample, now we will put this tube into the incubator at 45 degrees Celsius for 24 hours. On the next day, we will observe the acid and gas formation. If there is an acid and gas formation, then we see that E. coli, fecal E. coli, specifically fecal E. coli is present. If there is no acid and gas production, then we see that fecal E. coli is absent. Okay, this is the original tube, and here you can observe the color change. Now this purple color has been changed to yellow color, which means that acid has been produced. In the composition of the McConkey lactose bile broth, there is the lactose sugar is there. Now this lactose sugar has been fermented, converted into the acid and the gas. Due to the acid production, there will be the color change from purple to the yellow. Now here there is the Durham's vial is there. Here you can see there is the Durham's vial, and here also there is the same Durham's vial is there. Okay. Now here you can observe the little there is the bubble formation, which means that gas formation is also there. So we see that if there is the acid and gas production, if we observe like this, then we see that fecal E. coli is present and we consider the positive, which means that in the given water sample fecal E. coli is present. If we do not observe this kind of tube, acid and gas production, color change, then we see that the test fecal E. coli that is absent. This is for the McConkey lactose bile broth. If we are taking BGLB, brilliant green lactose bile broth, originally it is green in color. Okay, if fecal E. coli is present, then it will convert, that will ferment the lactose in the brilliant green lactose bile broth. There is a lactose sugar that will ferment into acid and gas. If in the water sample, if it is the fecal E. coli is present, that will ferment those lactose which is present in the brilliant green lactose bile broth, and we will observe the color change like this from green to yellow. And we can also observe if there is a Durham's vial, there will be the bubble formation, which means that acid and gas production if we will observe, then we see that E. coli, fecal E. coli may be present. And if we do not observe, then we see that fecal E. coli is absent. And to perform the elevated temperature test, one thing that we have to remember, first of all we have to take a tube and we have to add a sample, and in the incubator we have to incubate at 45 degrees Celsius. 45 degrees Celsius, that we don't have to forget, because normally we used to incubate at 37 degrees Celsius, but here we have to incubate this tube at 45 degrees Celsius for 24 hours. And on the next day we have to observe for the acid and gas production. Okay, for the elevated temperature test, it is specifically for the E. coli. We should know about the what is coliform. Coliform bacteria are the organisms that are present in the environment and in the feces of all warm-blooded animal and the human. This coliform group of bacteria that are present in the feces of all warm-blooded animal including human. However, the presence in the drinking water, if there is a presence of in the drinking water, that indicate that the disease causing pathogen that is present. The most recent development in the evolution of the coliform test is the ability to specifically identify and quantify E. coli in a water sample through the use of elevated temperature test. Through the use of elevated temperature test, we can identify and quantify fecal E. coli that is present or not. The presence of E. coli is as much more certain indicator of fecal contamination that is the presence of fecal coliform. Okay, if there is the presence of E. coli, fecal E. coli, it is an indicator of that somehow in the water sample there is the contamination of the fecal coliform, specifically E. coli. Okay, fecal coliform, how we should know about the what is coliform and what is fecal coliform. Fecal coliform, it is a subset of coliform group of bacteria. They are the subset of the coliform group of bacteria that are able to grow at 44.5 degrees Celsius. They are the thermotolerant coliform. They are thermotolerant. Normally, we incubate our tubes at 37 degrees Celsius, but here we are incubating our tube at 45 degrees Celsius because this coliform group of bacteria are the thermotolerant. Fecal coliform are the thermotolerant coliform. E. coli is able to grow at this temperature while most of the coliforms of limited sanitary significance do not. This elevated temperature test is used to differentiate between the typical and atypical coliform. Typical and atypical coliform. Typical, which means E. coli, and atypical means Enterobacter. Okay, it is used to differentiate between the E. coli and the Enterobacter coliform is on the basis of the growth at elevated temperature. E. coli is able to grow at a temperature of 44 degrees Celsius, or we can say that 45 degrees Celsius, while the growth of Enterobacter is inhibited at this temperature. Enterobacter will not grow at this temperature. Only the fecal E. coli can tolerate this temperature. So that's why we say is that fecal coliform, they are the thermotolerant and they can ferment the lactose into acid and gas. The things that we have to remember for the fecal coliform, the presence of fecal coliform indicate that the presence of fecal material from warm-blooded animal. Fecal coliform are the thermotolerant coliform, including all coliform that can ferment lactose at 45 degrees Celsius. Okay, what is coliform? Coliform bacteria are the organism that are present in the environment and in the feces of all warm-blooded animal including human. This is present in the feces of man and the other warm-blooded animal. The presence of coliform bacteria in the drinking water may indicate that the possible presence of harmful and disease-causing organisms that are present. Coliform, this is what all about the coliform. Okay, what are the characteristics of coliform? Coliform bacteria, they belongs to the family that is Enterobacteriaceae, which can ferment lactose. Organism that comes under the coliforms are the Escherichia coli, Enterobacter, Klebsiella, Serratia, Citrobacter and many more. This all are the coliform group of bacteria such as E. coli, Enterobacter, Klebsiella, Serratia, Citrobacter. Collectively, this group are the gram-negative bacilli, which means that rod shape. They refer to as a coliform because they share the similar morphological and the biological characteristic. So, these are the characteristic of the coliform. They belongs to the family Enterobacteriaceae, which can ferment lactose sugar. That includes Escherichia coli, Enterobacter, Klebsiella, Serratia, Citrobacter. They are the gram-negative bacilli shape. They are referred to as a coliform. Okay, furthermore, description is also there. They are the facultative anaerobes. Coliform group of bacteria, they are the facultative anaerobes, gram-negative, non-spore forming. They are the rod shape, like this way, rod shape. They can ferment lactose, specifically lactose sugar, and that can ferment lactose and produce acid and gas. Ferment lactose specifically, okay. And these bacteria, the name is here, and they are commonly found in the contaminated. They are commonly found contaminated in water. They share the similar morphological, biological characteristic. So this is what all about the what is coliform, what are the characteristic of coliform, and what is the fecal coliform. And Eichmann test, or we can say that elevated temperature test, it is usually for the fecal coliform test. In this we check the fecal coliform in the water sample, is it is a present or not. For that we take a tube, that is BGLB broth and McConkey lactose bile broth, any one we can take, and we observe the acid and gas production by incubating those tube at 45 degrees Celsius.